Welcome once again. Welcome back to Facts in Biology, a YouTube channel that works for the preparation of NEET exam and medical entrance exams which are to be conducted in the next year. Friends, last few videos we are trying to go into the depth of reproduction in plants, lower as well as higher plants, in which almost we have completed the reproduction in lower plants, asexual reproduction, vegetative propagation, and then we have proceeded on to the angiosperms. In angiosperms, we have studied asexual reproduction, vegetative propagation, as well as sexual reproduction also in depth and detail. Now, once again, I would like to say a big welcome to Facts in Biology, a channel that is devoted for the preparation of your NEET preparation. So once again, welcome. So dear students, if you find these videos helpful for your preparation, for your studies, kindly do subscribe, like, share and comment on this YouTube channel titled Anand Joshi Biology, so that the notifications can reach your inbox and you can very easily get notified whenever there is uploading of a new video. Thank you. Friends, today we are going to discuss the concluding part of reproduction in lower plants and higher plants. And obviously the last topic, although it is small, but it is very important for your NEET preparation. Today we are going to understand the concept of apomixis, parthenocarpy and polyembryony. So friends, today the first concept that we are going to understand is apomixis. Now apomixis in botany, it was first defined by a botanist Hans Winkler, and according to him, apomixis is replacement of normal sexual reproduction by asexual reproduction and without fertilization. Mind it well friends, this is practiced by reproductive organs. These are practiced by reproductive structure, but that doesn't involve the fusion, that doesn't involve the meiosis, that doesn't involve the fertilization. And that's why it is characteristic feature of the angiosperm. In Latin meaning or in Greek meaning, the Greek etymology of apomixis can be understood as apo, that is no or away from, and mixis, that is mixing of gametes, mixing of chromosome, that is we can say occurring during the sexual reproduction. So it is a kind of reproduction which is practiced by organs of the plant which are basically meant for sexual reproduction, but due to some changes, due to some morphological, physiological changes, instead of sexual reproduction, they practice asexual reproduction, and the process is called as apomixis. Now, apomixis is the phenomena of formation of embryos through asexual reproduction without formation of gametes and without the act of fertilization. Alternately, it is an unusual sexual reproduction where there is no meiosis, there is no syngamy, there is no fertilization. Embryo develop in the ovule and ovule develops to form the seed. So basically here the process of gamete formation as well as fertilization are completely abundant, and without these processes the reproduction is practiced, and such a reproduction is said to be the apomixis. In apomixis, when a gametophyte, organ or cell produces embryo-like structure without fertilization, it is said to be the apogamy. Mind it well, in apomixis, when a gametophyte, organ or cell produces embryo-like structure without fertilization, it is termed as apogamy. Similarly, when diploid sporophyte or sporophytic cell produces a diploid gametophyte without undergoing meiosis, without undergoing reduction division, it is said to be the apospory. So in this particular slide, you understood the meaning of apogamy and apospory. Generally, these are found in the plants like oranges as well as mango plant, they show apomixis and apogamy. The phenomena of apomixis is actually belonging to three categories. The first one, it is called as recurrent apomixis. The second one, it is called as non-recurrent apomixis. And the third one, it is said to be the adventative embryony. The first apomixis is recurrent apomixis, and in this type, the embryo sac generally rise either from the archaeosporial cell or from some other cells, other part of the nucellus. In diplospory, the unreduced embryo sac is actually derived from diploid megaspore mother cell, which is supposed to undergo meiosis, but that mechanism of meiosis fails, and as a result of that, without meiosis, that diploid megaspore mother cell is developing into the embryo sac, and generally it is found in the plants like Taraxacum. In apospory, the nucellar cells give rise to apomictic embryo sac. Mind it well, these apospory, it results into formation of nucellar cells, and these nucellar cells will give rise to what we call it as an apomictic embryo sac. The second type of apomixis is non-recurrent apomixis. In this type, megaspore mother cell undergo usual meiotic division, reduction division, and a haploid embryo sac is formed. Now, this haploid embryo sac, it will give rise to embryo either from the egg or oosphere by the process of parthenogenesis or parthenocarpy, or from some other cell, other haploid cells of the gametophyte, and that is through the apogamy. Now, due to apomixis, non-recurrent apomixis, when plants are produced, now these plants which are produced by non-recurrent apomixis, these are generally sterile and they are unable to reproduce sexually to produce next generation. So termination of, we can say, genetical material, it is expected in this non-recurrent apomixis. Generally it is found in plants like Nicotiana tabacum, the tobacco plant. The third type of apomixis is called adventive embryony. Friends, in this type, embryos may develop from somatic cells of nucellus which are diploid, or integumental cells which are again diploid, along with the normal zygotic embryo. Now very importantly here, the normal embryo is developing, it is continuing its growth, along with it additional adventive embryo also develop, and this additional adventive embryo, it may develop from the somatic cells of nucellus, or it may develop from the somatic cells of the integuments. And this particular type, or this adventive embryony, it is common in plants like mango, oranges, or even the lemon. And this gives rise to a condition called as polyembryony, because within the single seed, the reproductive embryo, sexual embryo, as well as non-reproductive embryo, or we can say somatic embryo, both of them are present, and this condition will definitely lead to presence of more number of embryo within a single seed, and that is called as polyembryony. And we are going to discuss this in little bit of time. Basically, if we see apomixis, generally identical plants, they can be produced effectively by the process of apomixis, very effectively and rapidly too. The second concept that today we are going to discuss in this video lecture is parthenocarpy. Now just now we have discussed a little bit of parthenocarpy, that development of an egg without fertilization, it is called as parthenogenesis, or it is called as parthenocarpy. Friends, this term parthenocarpy was first coined by Knoll in 1902, and it is a condition in which fruits are developing without the process of fertilization. So not only the embryonic development, but the ovarian development is also occurring without fertilization to form what we call it as a fruit. And it occurs normally in some of the varieties. Naturally, it occurs in some natural varieties of pineapples, banana, as well as in papaya, where you can have parthenocarpic development naturally without artificial efforts. Now in these plants, it seems that potential placental tissue in the unfertilized ovary, it produces a plant hormone auxin. We call it as an indole acetic acid, IAA, which is responsible for enlargement of ovary into the fruit without fertilization. Now the fruit resembles the normally produced fruit, but it is without the seeds, it is without the embryos, and that's why it is many times referred as seedless fruit. Now here you can summarize the parthenogenesis or parthenocarpy. Now here what we call it as a normal mature ovary is there. Now this mature ovary, it may show hormone dependent polyembryony or it is a parthenocarpy. Now this is hormone dependent. When hormones are there, when hormones are naturally produced like indole acetic acid or auxin, it may result into the abortion of fruit as it is an abnormal process, or it may lead to the fruit expansion without fertilization. So we can obtain the seedless fruit here. Additionally, if we see this mature ovary, it may have hormone unassociated, not related to hormone. And here the regulation of this parthenocarpy is with the help of some specialized protein that are called as 19NP. Now these 19NP, again they may lead to the hormone independent parthenocarpy, and this hormone independent parthenocarpy, it may lead to the dormant state of the fruit which is not going to develop normally, or in another way that may develop into a parthenocarpic fruit, or what we call it as an parthenocarpic seedless fruit. So this is the way either hormone dependent or hormone independent mechanism is there, where the development of ovary either it is terminated or it is paused, or it may proceed on to the formation of seedless fruit or parthenocarpic fruit. Friends, the third concept that we are going to study today, it is nothing but the polyembryony. Now this polyembryony, it can be seen in this particular slide. You can see the photograph where the seed has been taken into a vertical section, and here you can have a normal embryo which is located here, and in addition to that, these are the somatic embryo which develops without fertilization. So here a single seed, it may contain more number of embryos, and that's why it is called as polyembryony. Actually, polyembryony, it is the development of more than one embryo inside the seed, and the condition is described as polyembryony. Now this polyembryony was first observed by Leeuwenhoek in 1719 in the seeds of citrus, or that we can say belongs to lemon family. Now it is the occurrence of more than one embryo in a seed which consequently results into emergence of multiple seedling within the single fruit. The additional embryos results from differentiation of or development of various maternal zygotic tissue associated with the seeds. This polyembryony may be true or false polyembryony depending upon whether many embryos arise in the same embryo sac or in the different embryo sac in the same ovule. So a single ovule may have more number of embryo sac, that may also result into polyembryonic condition. In adventive polyembryony, an embryo develops directly from the diploid cell of nucellus and integument in the plants like citrus. In cleavage polyembryony, zygote proembryo, sometimes it divides or cleaves into many parts, and these parts consequently develop into the embryonic structure. Now each unit of them, it will develop as a separate embryo. Now polyembryony increases the chances of survival of new plant, because if one embryo fails, the other will develop and there will be the formation of new plant. Now nucellar adventative polyembryony is greater importance in modern horticultural practices, because today we require more amount of production from the plants, more amount of greater we can say benefit from the plant, so that polyembryony can be commercially exploited, utilized in modern day agriculture. So this is the story of last topic of the chapter, that is consisting of apomixis, parthenocarpy as well as polyembryony. And if you find this video helpful for your preparation of NEET examination, kindly subscribe, like, share and comment and strike the bell button to the YouTube channel Anand Joshi Biology. For the time being, it's goodbye from me, Anand Joshi Biology, a YouTube channel that is devoted for your preparation of NEET examination. Thank you friends. Thank you very much.